Oh, thanks, Kathy. And recording. All right, greetings everyone on Zoom. Greetings out there on YouTube. Thanks for coming. This is our second to last Water, Wetlands, and Watershed Seminar for fall, spring 2025. And we're really happy to have our own colleague here, Dr. Chamtot O, or Cham. He's in the room sort of there over on Zoom and was not feeling well today, so he's presenting virtually. So I'll give a brief introduction and then Cham, the floor is yours. So Dr. O is an assistant professor in the Department of Environmental Engineering Sciences here at the University of Florida. He has his BS and MS degrees, both from Seoul National University in South Korea, and then he has his PhD from the University of Illinois at Urbana-Champaign. And as we'll hear about today, Dr. O's research focuses on environmental genomic surveillance, and we'll understand a little bit more by the end of today what that means, and microbial water quality. His work aims to better understand the fate and transport of pathogens, antimicrobial resistant genes, which a lot of folks are getting more and more, we'll say, interested in nowadays, and microbial interactions in engineered and natural water systems. And so maybe as you're listening today, either online or here in the room, try to think about what are the differences between engineered and natural systems and where they might cross over. He's also leading efforts to develop monitoring tools, so like rapid online monitoring tools for real-time microbial surveillance in water infrastructure, which also we've been hearing a lot more about ever since basically COVID tracking in the wastewater supply. So Cham, the floor is yours, and have at it.
Thank you very much. Hello, everyone. Hi, everyone. So I have a cold. I really wanted to make it in person, so please forgive me not presenting there or my unpleasant voices. So what I wanted to present today is about environmental genomic surveillance. And this is quite an emerging research tool. And I want to introduce what this new tool is and how we can utilize this tool to address environmental issues. So this is outline of my talk today. I first introduce environmental genomics surveillance system, and then I will move on to introducing my ongoing research topics, as my goal today is to introduce environmental genomic surveillance. So I will briefly skim through different research topics rather than focusing on one topic in depth. I want to highlight what types of environmental issues we can address with this emerging research tool. So my ultimate research goal is to mitigate the impact of microbial activity in the environment on public health and the economy. And you can name numerous reasons how microbes affect our life, but I like to present the two reasons. So pathogens can contaminate recreational water, such as river or coastal water, or contaminate drinking water sources that result in pathogen infection. And microbes also are designed to survive and thrive at where they are. So for that purpose, they do metabolic activities. And these activities transform chemical species of substances. And because carbon and nitrogen are important nutrients for the microbes, they play a critical role in global carbon and nitrogen cycle. Then how can genomic surveillance help us understand those microbial activities? I'd like to explain what microbial activities are with these illustrations. So all living creatures have DNA. DNA consists of hundreds or thousands of genes, which are depicted in red piece. And each gene can be transcribed into mRNA, and this mRNA can be used to translate proteins. And what we call microbial activity is actually the result of group activity of proteins. So microbial activity involves the presence of DNA and transcription of mRNA. All microorganisms have one or few DNA, and the number of DNA remains stable throughout their life cycle. On the other hand, mRNA level can vary significantly depending on environmental conditions. When microbes are under certain conditions that result in doing some activities, then what microbes do is transcribe mRNA. And if the situation changes and they need to reduce or stop the reactions, then they degrade mRNA. So DNA can represent microbial abundance in the environment, and RNA can represent how microbes respond to the environmental change conditions, which I call microbial activities. With that, I'd like to define environmental genomic surveillance as systematic collection of environmental samples and analysis of DNA and RNA in order to track the fate and activity of microbes in the environment. Environmental genomic surveillance involves many challenges. I like to compare environmental genomic surveillance to clinical genomic surveillance that perform genomic analysis on clinical samples. Clinical samples are from mostly one host, whereas environmental samples are mixtures of everything, which looks like this. Now let's assume the colored microbes here are our target. In many cases, our target microbes present at low prevalence, and they are genetically diverse. This means even though they are in one species, they are not identical, like we are all Homo sapiens, but are not identical. On top of that, our target microbes exist together with complex microbial communities. So it's challenging to define our target microbes. Here, green things in the illustration present impurities such as organic matters or ions that could interfere with molecular analysis. More importantly, our target environmental field lies on a large area, so we have to strategically take samples to understand the spatial dynamics. I believe environmental engineers and scientists are well qualified to track these challenges because basically what we are doing is to understand fate and transport of matters in the environment. We've learned and studied physiochemical reactions in the environment. For example, we understand surface charge of particles and the absorption-desorption reactions, which is the key information to understand the interactions with other particles. And we know how hydrological conditions decide like whether or not certain particles flow through the streams or settle down to the bottom. We also know weather and climate conditions affect these substances. Furthermore, we can think all these reactions on different scales from micro to a global scale. And I believe environmental genomic surveillance is simply replacing our target matter with genomic materials like DNA and RNA. So what I want to emphasize on this slide is, although we are not a microbiologist, but we can make unique contributions to this field of study. Now let's move on to next chapter. I will show you how these unique contributions look like. The first research topic is about improving environmental genomic surveillance itself. And the other three will be presented to show what environmental issues we can address. Environmental genomic surveillance involves roughly these three steps. First, for genomic surveillance, someone has to go out to the field to take samples, and they need to bring this sample back to laboratories. And with this sample, we have to increase our target genetic materials and reduce impurities. Once this processing is done, the samples are ready for molecular analysis. Researchers, including myself, has made an effort focusing on improving the sample processing and analysis of our target DNA and RNA. However, based on my experience, going out to the field to take samples would be the most challenging and time-consuming, labor-intensive works. And I personally tired of sampling as well. So what I've been imagining so far is to develop a certain system that we can install in the field. And that system can take samples, process, and analyze samples on site, and just send the data back to me. Because I'm a big fan of big data, And I believe as long as we have high resolution spatial temporal data, with an aid of AI technology, we'll be able to better understand what's going on in the environment. And this is the simple design of what I have in mind. And first module will be designed to take sample from environment like coastal water or river to take microorganisms. And then it's designed to take DNA and RNA out of the cells, which I call microbial lysis. And these RNA and DNA will be isolated from all impurities and purified in the absorbents. And these purified genomic materials will be conveyed to the second module, which is PCR. And the basic approaches to this system is to taking a large volume of raw samples. If you have to bring your sample to laboratory, your sample volumes are limited to like 1 liter or so. But because we'll install this system on site, we can process the water however we want, like 100 gallons if we need it. And we'll design processing method as mild as possible, such as heating the water with salt, which is sufficient to discharge the processed sample back to the site. Although these mild processes will have a decent DNA and RNA recovery efficiency, we expect the lower efficiency will be compensated by the large volume of the input sample. And this rough calculation about the processing efficiency tells us if the first mild purification process can recover 10% from large volume of the sample. And if needed, we can add advanced technology to improve the efficiency. And if we achieve a 50% from the second process, the result concentration will be 10,000 times higher than the raw sample. And we are currently working on this PCR module. So this is the PCR design of our PCR. PCR has pretty simple design. It has thermocycler and fluorescence measurement. And as shown in this drawing, there is a reactor in the center, which is surrounded by a heating block. And this copper block is attached to the Peltier module to control the reaction temperature. And we have excitation light source here and emission light detector on the bottom. And the temperature and the fluorescence can be monitored as shown in the photo and the image on the right-hand side. We're still optimizing the prototype of this PCR, but what makes this PCR system different from conventional PCR is this will be automated continuous flow PCR system. What I mean is the reactor here will be connected to the programmable pump so that we can convey the exact volume of PCR reagent and nucleic acid purified from the first module at the determined time frame. So this PCR system doesn't need personnel to operate the system. All right, let's move on to other research topics. I'd like to introduce what types of environmental issues we can address with this environmental genomic surveillance. First topic is about tracking the spread of antimicrobial resistance genes due to hurricanes Helene and Milton. If you still remember, these two hurricanes made landfalls in Florida last year, just 2 weeks apart. After Helene, we tried our best to clean up the debris and pollutants, but the cleaning capacity was its max, and many of the garbage or pollutants were just temporarily piled up without being properly secured. And Hurricane Milton just redistributed what we piled up, and which ended up in coastal water. And Center for Coastal Solutions under the leadership of Dr. Angelini found this situation serious. And we went out for taking samples ranging from Tampa Bay through the Punta Gorda as shown on the map. And what my laboratory found with these samples are the elevated enterococci levels, which indicates the fecal contamination. So this is the evidence showing the untreated wastewater and non-point source pollutants were discharged from inland. And what this situation makes worse is the outbreaks of Vibrio vulnificus, which shows the high mortality, like 20%. Florida Public Health Department reported doubled cases of Vibrio vulnificus when the Hurricane Milton hit the Florida. Through the collaboration with Dr. Morrison and Dr. Jung in the Department of Environmental Engineering Sciences, we hypothesized this situation. The freshwater bacteria that can contain high abundance of antimicrobial resistance genes, which is denoted in the red circle, can form biofilm on microplastics, which will confirm the elevated concentration. And once this bacteria can form biofilm, and this biofilm could be the place where antimicrobial resistance genes are transferred to indigenous marine bacteria. If that happens, antimicrobial resistance gene could establish in the coastal water, and the negative impact from the storm surge due to the hurricanes can last much longer than we initially thought. So fortunately, NSF bought our hypothesis and support our sample analysis. And this is what we're gonna do with these samples. Our goal is to find the evidence showing horizontal gene transfer within our samples. Horizontal gene transfer allows a piece of genes to be transferred to other microbes. However, there will be another mechanism of gene transfer, which is called vertical gene transfer, where the whole genome will be replicated and inherited in the same microbial species. So we'll examine antimicrobial resistance genes with identical sequences like this red color. And then we'll look into the adjacent regions. And if the adjacent regions are also identical, then this is the evidence for the vertical gene transfer. And if the adjacent regions are not identical, while having the same AMR, then this will be the evidence for horizontal gene transfer. We just initiated this project, so I hope to share some interesting results soon. Next topic is about nitrous oxide production in karst aquifer. It's been 1.5 years since I moved to Florida, and I'm so happy with such a beautiful nature in Florida here. And I love spending time with my family at springs like Ichitakuni Springs in this picture. But these pristine springs, more specifically Floridan aquifer or karst aquifers, may be a hotspot for nitrous oxide. Nitrous oxide is third most significant greenhouse gases, and many research groups have reported high concentrations of nitrous oxide at karst aquifers. And this study has been collaborated by Dr. Sean Yoon in the Department of Geological Sciences. Let me briefly explain what could happen in karst aquifer. Karst landscape comprises about 20% of all ice-free landscape, and karst aquifer has soluble rocks such as carbonate, which easily dissolve by water. So the karst aquifers looks like porous media. And depending on the geological feature, they could form conduit, which is like a large water channel. If the water flow of the surface water increases, then the surface water can infiltrate into this karst aquifer through the conduits and the porous media and replace the groundwater in this karst aquifer. And because groundwater typically shows anaerobic conditions, while surface water shows aerobic conditions, the karst aquifer will go through frequent transition between aerobic and anaerobic conditions. And this is the microbiological nitrogen transformation pathway. When anthropogenic activities like using fertilizers at agricultural fields discharge ammonia and nitrate into water. And these nitrogen species go through these transformation processes. And these nitrogen species can be transformed to nitrite, nitrous oxide, and nitrogen gas, depending on the environmental conditions. So we want to learn how hydrological conditions like this affect microbiological activities as introduced here. We chose a Santa Fe river sink and rise system in Florida, which is about 1 hour from here. We believe this is ideal site to study the relationship between hydrological conditions and microbial activities, because as you can see in this map, Santa Fe River flow like this, but suddenly the river disappear at river sink. And what actually happens is the water flows through the underground conduits, as shown in yellow color. And the Department of Geological Sciences at UF have investigated this site a lot. And now we have clear understanding how conduits looks as shown in these illustrations. And the water forms a river again at the river rise. And USGS installed water stations at river sink and river rise. So we know the exact volume of the water that recharges in karst aquifer. And there are 2 monitoring sites which we can analyze the water quality in the karst aquifer. There was a quite big rainfall in the past January, and we went out for sampling throughout the rainfall event. Here, the black solid line shows the surface water flow rate to aquifer. So if the line is above 0, this means the surface water fills the karst aquifer. So we confirmed this rainfall event induced a surface water infiltration into the karst aquifer. We also measured the nitrous oxide during the rainfall event. And we found a similar trend with the time lag. And what this means is the karst aquifer conditions changed from anaerobic to aerobic, and this induced the promoted nitrous oxide production. And you hypothesize that this is because mild aerobic conditions disrupted this microbial transformation pathway from nitrous oxide to nitrogen gas, which is called incomplete denitrification. So we analyzed microbial community through 16S rRNA and 16S rDNA sequencing. And this PCA analysis shows how microbial community changed during the rainfall event. This W31 data points represent initial microbial profile, and the microbial profile shows significant change throughout the rainfall events. And the last sample, W36, represent the microbial profile at the last sample, shows the microbial characteristics return back to the initial stage of the microbial profile. And this data indicates rainfall events somehow impacted microbial activity and abundances. And we also quantified this NIR and NOS gene. And if the NIR over NOS gene is large, that means the nitrous oxide production is dominant pathway. And if the NIR over NOS is small, then nitrous oxide consumption is the dominant pathway. And here, this graph on the bottom shows NIR over NOS ratio over time. And as you can see, the trend of this ratio followed the trend of N2O production. So we believe this is the evidence showing the microbial activities indeed caused N2O production. Now that we know microbial nitrogen transformation is the key mechanism for nitrous production in karst aquifer, we wanted to evaluate these microbial activities in a more quantitative way. Dr. Yoon and I are working on developing biomicrofluidic system to unveil microbial N2O production mechanisms. This system is basically a microfluidic system where we can fabricate any designs of chips so that we can mimic different karst aquifers. And we have two water channels that represent surface water flow through the conduits and groundwater flow through the karst aquifer. That what my laboratory is doing is to design bacteria to produce green fluorescence when nitrous oxide production is the dominant pathway, and produce red fluorescence when nitrous oxide consumption is the dominant pathway. And we'll grow this bacteria on this microfluidic system, so we'll see the color change depending on the hydrological conditions. And with that data, we'll explore how hydrological conditions affects microbial activities. All right, the previous two topics were about microbes in the nature. And this last topic is about microbes in the engineered system. The title for the last topic is monitoring biofilm dispersion from the drinking water distribution systems. And this study has been collaborated with Dr. Hyun-Suk Oh from Seoul Tech. There were at least 200 drinking water associated outbreaks in the United States from 2015 to 2020. This may be a huge number if you think we are living in one of the most developed countries. And I'm confident with the water treatment technology, and I would be happy to drink the water discharged from the drinking water treatment plants. However, when they convey the water to each household, the water passes through the drinking water distribution system where there are biofilms in it. And these biofilms are biomolecular complex where microbes, including pathogens, can thrive. And these biofilms protect microbes from residual disinfectant. So pathogens that present at low level when they discharge from the drinking water treatment plants can increase the concentration in the drinking water distribution system to the level they can infect human beings through drinking water consumptions. Because taking sample from inner surface of the pipe disrupt water supply, it's not easy to regularly monitor biofilm in drinking water distribution system. So our approach to address this issue is to analyze microbes in the tap effluent. But let me briefly introduce biofilm life cycle in drinking water distribution system. The planktonic bacteria from water treatment plants can bind to the inner surface of the drinking water distribution system, and they produce sticky chemicals that strongly bind themselves to the pipe surface. And then biofilm can start growing from the surface, and once they are matured, then bacteria can release back into the bulk solutions. And if we analyze microbes in the tap water, there will be the planktonic bacteria from water treatment plants and bacteria released from the drinking water distribution system. I will call planktonic bacteria and this as a biofilm bacteria. just for the convenience. So our research question is, how can we differentiate biofilm bacteria from planktonic bacteria in the tap effluent? Our approach is to focus on quorum sensing process, which is bacterial communication mechanisms. So believe or not, bacteria can communicate each other. Bacteria produce these quorum sensing signals to the surrounding environment, and at the same time, they can sense the concentration of these chemicals. And these chemicals upregulate the quorum sensing reactions, meaning they can produce more chemicals when they detect the quorum sensing signals. And once this signal exceeds a certain level, then they initiate the downstream gene expressions, including the biofilm formation. So quorum sensing is population density dependent microbial reactions, and it's involved in biofilm formation. Cell density varies significantly depending on two bacterial modes of growth, which are planktonic bacterial growth and biofilm growth. On the left-hand side, you can see the planktonic bacteria can replicate in bulk solution. But cell density increase is like just changing from rural area to suburban areas. But biofilm growth is like complex apartment in megacities. The cell density of biofilm bacteria is 2 to 3 orders of magnitude higher than that of planktonic bacteria. So we hypothesize the quorum sensing related mRNA will be produced more in this bacteria compared to planktonic cells. And we chose last eye gene, which is because this is well studied. This is one of the quorum sensing related gene. To test our hypothesis, we grew bacteria in 2 reactors with different specific surface areas. The small reactor has 18 times higher specific surface area than the large reactors. And we analyzed lost eye mRNA in the cells from these 2 reactors. Because the planktonic bacteria grow in bulk solution, while biofilm bacteria grow on surfaces, we assume the small reactors will have higher impact from biofilm bacteria. And the result shows that the bacteria from the large reactor, which is shown in the red circle, did not show a significant loss site gene expression as population density increased. However, small reactors where it has more biofilm impact showed a loss site gene expressions. We also grew bacteria in tubing system under flow conditions, and we flew tap water through the tubing with biofilm. What this data on the right-hand side shows is the last eye mRNA in the effluent shows a strong correlation with the mRNA of the biofilm in the tubing. So given all together, what our study shows is that we can investigate biofilm dispersion from the drinking water distribution systems by analyzing the quorum sensing mRNA in tap effluent. So I've just skimmed through all our ongoing research. If the presentation was too busy, I'm sorry about that. But through this seminar, what I wanted to introduce is environmental genomic surveillance and what type of environmental challenges we can address with this tool. So this is the take-home message for you. Investigating DNA and RNA in the environment helps us understand how microbes impact both environmental and public health. So this is all I prepared. I'd like to shout out all type of supports for our study, especially all students from my laboratory who are highly motivated and they work really hard. Thank you very much. If you have any questions, I'll be happy to take from audiences, but you can reach out through different channels for further discussions. Thank you very much.
Thank you, Chem. Thanks, Dr. O, for sharing with us despite your voice and
I'm
everything.
so sorry.
It was very clear. So we'll start with some questions in the room and then we'll check on YouTube. I know many of our students are watching on the YouTube stream right now, so please pop your questions in the chat there. We'll start with questions from the room. Okay, I'll ask a question. Okay, so Chem, the study that was most relevant to the work I've been seeing recently is the nitrous oxide production in the aquifer, the N2O. And I think one of the major challenges we have when we think about nutrient pollution at the land surface, and especially after it recharges the aquifer, and then maybe 1 year, 5 year, 30 years later emerges from the spring. And we want to understand what the load of nitrate from the land surface, how much is denitrified along the pathway, and even how do we model it? First order, zero order, second order. Is any of the work that you're doing right now able to help us understand what are basically the half-life, something simple like a half-life or a denitrification rate constant, an area or volume weighted removal rate constant that we can use because not every molecule of nitrate that enters the aquifer leaves, and some portion is denitrified and is not becoming pollution for downstream water resources.
Right, great question. So if I understand your question correctly, the water flow underground will show different time scale. And actually, the Department of Geological Sciences has investigated a lot about that. And what they found is the relationship with the isotope analysis. What they found is the nutrients of carbon or nitrogen that infiltrate into the aquifer will show us like decade or from decade to like days. If the geological feature allows the water coming out of the aquifer through the spring, then they could impact us like within days. But if the geological feature allows the water flow through the long journey through the karst aquifer, then the impact will come out after like decades. So it highly depends on geological feature. And the reduction mechanism is mostly because of the microbial activities. And that microbial activity also depends on the hydrogeological feature that I just described on this slide. So it really highly dependent on the environmental conditions.
Right. So assuming we know the travel time distributions, which we often do, at least through models, right? So we have the overall proportion of water that spends 1 week, 1 day, 10 years, 100 years. So we have the travel time distribution, and we want to basically apply some kind of removal rate coefficient to, if it spends a week, it has a shorter time, but the removal rate constant is whatever, negative 3.7.
Right.
And if it spends 100 years, it's a longer time, but the removal rate constant is still first order negative, same number. So what I'm looking for is like, how can we get a better feel for the spatial variation in overall capacity of the aquifer?
If we assume the primary mechanism for the nitrous side transformation is microbial activities, that microbial activity really depends on the environmental conditions. So what we wanted to reveal with this biomicrofluidic system is we want to provide more quantitative way to evaluate the reduction of these denitrification processes. Because so of course we can average all the hydrological processes like with the average precipitation, but the precipitation events really depends on case by case. So what we are going to explore through this system is the strength of the rainfall event and how that strength affects with the geological feature like conduits and porosity of the porous media um, collectively affects the microbial activities.
Okay, thank you, Chem. I want to follow up with you on about that a little bit later, some potential ideas. Other questions from the room? YouTube is very quiet. Okay, if no other questions, then let's give Dr. O another round of applause and let him get to bed or take a nap or something. Thank you so much, Chem. We appreciate
Thank you
you
so much for having me here.
presenting despite the illness. So next week we have our final seminar of the semester. We have Dr. Biogeochemical is coming in. I believe he's going to be presenting virtually as well. One moment, let me just check to be sure. I've caught up, if you're in the environmental engineering sciences section, I've mostly caught up on the grading. I know there was some confusion with the due dates and some of them were like from in the deep past. And so that's all updated. If there was confusion about what you uploaded for which week, don't worry, I can see them all. And so I've gone through and graded those. Yes, so next week we have Hymenontate Biobill from Ag and Bioengineering. And I think he's going to be here in person. So we'll see you all next week. And until then, be well. Thanks, everyone. Cheers.
Thank you.